To my poster presentation on the characterization of antifungal resistance in pathogenic Candida to improve the development of resistance biomarkers and species identification. My name is Candida CzajkaK. I have no affiliations to declare, and this poster session has received financial support from the NOMA AFP Innovation Fund, the NASAMU Faculty Association Research Development Award, the NHRC Internship Fund, the NSERC Discovery Grant, and Northern Cancer Foundation in the form of educational and research grants. Candidiasis is a highly pervasive infection posing major health risks globally, especially for elderly, immunocompromised, and palliative populations. Pathogenic Candida species have evolved intrinsic resistance, which is an innate feature of that species, or they have acquired antifungal resistance by gain-of-function mutations in target pathway genes, drug efflux pumps, or transcriptional regulators. The four main drug types include the azoles, paulines, echinocandin, and nucleoside analogs. The objectives of this study were to identify and characterize antifungal resistant samples from a set of clinical isolates retrieved from patients with oral candidiasis infections, as well as a second objective is to develop point-of-care testing to identify the species of these infections and develop biomarkers. So about 80 samples were retrieved from patients aged 65 or over at the time of antifungal prescription and 2 weeks later after treatment. The strains were tested for susceptibility of different antifungals using the standard E-test and broth microdilution methods. The strains were further cultured in growth curve experiments to measure their growth with spectrophotometer readings of the absorbance at 600 nanometers, and this was done over a 48-hour time period. And various antifungals were tested, including nystatin, the echinocandin caspofungin, and 4 azoles. DNA, RNA, and protein was extracted from the samples when they were growing at mid-log phase and stored for future analysis. The results include a predominant Candida albicans among the sample set, as well as Candida glabrata and a few other species. Fluconazole was the first-line treatment prescribed in most cases. From the sample set, we found 8 resistant samples. Primarily, there were 5 Candida glabrata, and then there was 1 albicans, 1 tropicalis, and 1 cruce. For the growth curve experiments, we saw evidence of the intrinsic fluconazole resistance in the Candida cruce sample. We found acquired fluconazole resistance of the Candida tropicalis, despite this species being typically susceptible. And we found acquired echinocandin resistance, as well as intrinsic fluconazole resistance for the Candida glabrata sample. So for the future direction, this intrinsic fluconazole resistance of some species would be helpful to identify at the time of point of care. So the work is to develop an assay that can detect these species, as well as sequencing the resistant strains to identify mutations associated with the acquired antifungal resistance. And these validated resistance biomarkers could also be targeted for detection. The point of care testing assay is based off of a general microbial detection that is in development using LAMP PCR and CRISPR for a simple lateral flow readout. Thank you for taking the time to listen to my presentation.